Okay, well, thanks everyone, and thank you very much, Vlad, for the nice introduction. Yeah, it was too bad. I was looking forward to hearing Bernard's talk as well, to hear what he's up to these days. But we're going to have fun nonetheless. I'm going to talk this morning about some experiments that we've done on motor neuronal gene expression. And first of all, let me say that it should not be allowed for me to stand up and talk in a symposium that has the word gene expression in it, because I'm not a molecular biologist by any means. But we've reverted to looking at gene expression in motor neurons to try to explain some of the physiological changes that we've seen in motor neurons over the years by looking at their biophysical properties. So that's the reason why we're going to talk a little bit about gene expression today, as opposed to what I usually ramble on about, and that's the physiology of motor neurons and how they work. Of course, we're talking about the motor neurons that are in the lower spinal cord in the rat. We're not talking about motor neurons in the human, but I'm sure that this relates. And they're the cells there down by the, let me see if this works. Oh yeah, down at this level here that are below or distal to the motion generating, the pattern generating systems in spinal cord. And this is a stain from alpha motor neurons from the spinal cord that are stained for heat shock protein. And you can see the nice cell body there. You can see the nucleus that's stained for nuclear yellow. So why have we gone into gene expression? We had a perfectly good system going, looking at the physiology of neurons. Well, for a couple of reasons. First of all, a lot of the biophysical changes we see in motor neurons have to be explained by changes in either the proteins that are governing these properties or in their modulation. And so we thought that we'd look at the proteins, see if the proteins are changing. For example, you can change the excitability of neurons by changing, the amount of sodium channels in the membrane, for example, or the amount of potassium channels in the membrane. So we started to think, well, it would be nice to measure the protein. Protein is difficult to measure in small samples of motor neurons for a number of reasons. They're small. Some of the techniques we're using we're capturing small samples, and we have to combine them to have enough tissue. The second problem, probably more important, is that a lot of these proteins don't end up in a nice localized spot where you can measure them. They end up in the periphery. They end up down at the neuromuscular junction. They end up in the dendrites. And I'll show you examples of that in a minute. So to measure the protein changes in these neurons with things like exercise training is really quite problematic. So we thought, well, if the protein's changing, probably the mRNA is changing, and that would occur. We could measure that in the cell body where the proteins are made, and that would give us some idea of what's being stimulated, what genes are being stimulated. We do recognize there are limitations from jumping from mRNA to protein. There's not always a corresponding increase. When mRNA increases, it doesn't mean protein increases, and protein can increase without mRNA increasing. And we know all that, but we thought that it would give us some insight into how certain genes are stimulated by exercise, and it was a beginning. So forgive me if the molecular biologists in the audience, forgive me for my naivete in assuming some things. This is just an example. The 5-HT2A receptor, it's a serotonin receptor in motor neurons, and it is a protein that is found in the proximal dendrites and some in the distal dendrites. So it's spread a little bit in the cell body, proximal dendrites, distal dendrites, and it's a receptor that's very important for rendering the motor neuron more or less excitable when serotonin is around. Now this is an in situ hybridization, which means it's like immunohistochemistry, but it shows the mRNA instead of the protein. And you can see here that the 5-HT2A receptor mRNA is in the cell body. where you think it would be. So it allows us to measure, if this gets darker or lighter, it would mean that the gene was either more or less stimulated. And this is, of course, the sense, which shows that this is specific to the probe that we used. This is just a bigger amplification. You can see the cell body. Some of them you can see a nucleus. This is all mRNA, and this is a stain, a dab stain. And this is the sense. Same with 5-HT2C. There's the mRNA. Higher magnification. So if we can measure the mRNA in the cell body, we can get an idea of whether a certain protein is increased or decreased in its expression, and this might give us some idea about what exercise or spinal cord injury or whatever we're looking at, aging, is doing to protein synthesis. Okay, so this is a summary slide of some of the things that we found. This embodies several publications, which shows the physiological changes in motor neurons. So this is what really stimulated us to start to look at gene expression. All of these properties, voltage threshold, the rise of the spike, the amplitude of the AHP, all of these things change or are different between a highly active and an inactive neuron. And so, our thought was, well, I wonder if some of these changes are due to changes in the synthesis and incorporation of the proteins which lie under these properties. And I'm not going to talk about the physiology today. I've blabbed on about that before. Our thought was that this is what happens with exercise. And I should point out that, the spinal motor neuron is an example of a neuron. This kind of thing probably happens throughout the nervous system with exercise, and it could involve other neurons in the central nervous system as well. Motor neurons make for a nice model for a number of reasons, but anyway, so this was a model that we had proposed many years ago. It could be, go back. It could be neurotrophins that are being released by the target, which cause these biophysical changes, probably through a change in gene expression. Some metabotropic receptors, calcium coming in. Calcium comes in with each spike. So all of these things could stimulate phosphorylation events, which could stimulate some kind of change in gene expression and incorporate that into ion channels, receptors, which would change the biophysical properties of the neuron. So if you want to measure gene expression in motor neurons, you have to be able to have access to the tissue. And as you know, spinal cord is not just motor neurons, it's a whole bunch of other cells. It's interneurons, it's glia, it's all kinds of support cells. So years ago, we decided to use what's called laser capture microdissection to pick out motor neurons out of the spinal cord. So we could collect that tissue and look at gene expression by measuring the mRNA. So this is, we collected lumbar motor neurons. These are the motor neurons that innervate the legs. We used an Arcturus system. We're now using a Palm system, same principle. We took individual motor neurons in longitudinal sections. So we take the spinal cord, freeze it, slice it very thin, 10 to 12 microns. In those sections, you can identify motor neurons, usually just by their size. And you collect a whole bunch of motor neurons and do gene expression on the collected sample. So this was a study that, and actually these results have been published about 4 years ago in Neuroscience. And we did all the original studies to determine how long could we collect samples before we started to see mRNA degradation and so on. And it's about 30 minutes. So Lindsay was my first student to work on this, and she is using the laser capture microdissection unit. You can see a spinal cord section on there. That's a cross section in this case. She's looking for neurons. And in this particular experiment, we removed the lumbar cord. We cross-sectioned it. In her case, we cross-sectioned it. We fixed it, mounted it, stained it with Crescel Violet. And here's a picture of Lindsay here. She's now, I think, in medical school at McMaster. And basically, here's a cross-section of spinal cord. This is the ventral horn. And we would look for motor neurons. You can see them here. They're crescent violet stained. You can see the cell body and the nucleus. After spinal cord, after laser capture, you see the holes here left after we took them. Here are the neurons. There's the hole left. And you can see in this, these are some of the things that we've taken out. You can see here the neurons here. It's got a little bit of tissue around it, but at least we don't have other cells. You can see on a larger magnification here, there's the neurons that we've taken out. And then we take that tissue, we combine it, isolate the mRNA, or isolate the RNA, look at the integrity, do the reverse transcription, preamplification, and do PCR. And that's another advantage of this technique for us, is that you isolate motor neurons only, and also you can amplify the signal, which is not the case with proteins. So in this case, we can amplify the signal, and we could look at the mRNA content and assume to some extent that that means that gene was stimulated by exercise or whatever. Okay, so you're not expected to read this, but this is the study, first study that we did using laser capture, and we were interested in changes in motor neurons resulting from physical activity. And again, we did this because we had all these biophysical changes, and we wanted to find out what was causing these changes, what was being upregulated or downregulated. And so we measured a whole array of proteins here. You can see we've separated them out. These are ion channels, subunits, sodium, potassium channels. Here we've got neurotransmitter receptors. We've got some neurotrophin receptors down here. We've got some growth-related and so on. And we've got housekeeping genes down here. So we measured a whole array of things. And we were very disappointed with the results. We were able to publish it anyway. These were highly endurance-trained rats. They trained for 16 to 20 weeks, 2 hours a day, 2 times 1 hour. And we looked at the gene expression in a collection of lumbar motor neurons. Now, these were not motor neurons that were separated out by muscle or by anything. We just collected everything in the lumbar region that was motor neurons based on their size, as I said, which you can do, and their location. And these are the only changes that we found. Oops, no, go back. Oops. This is the biggest change we found. This is a receptor, a serotonin receptor. Very interesting result. A serotonin receptor that is found at the initial segment. It hyperpolarizes the membrane by modulation of sodium, that's supposed to be sodium channels. It tonically depolarizes the voltage threshold and prolongs the spike size. So what we found in our physiology was exactly, with training, we found some of these things actually changed in the direction you would expect if you downregulated the expression of this protein. So we found, we found hyperpolarization of the voltage threshold. We found a shortening of spike rise time. So this particular protein, which seems to inhibit or decrease excitability of the cell, was actually downregulated very, very significantly. And you're going to see later that this is a protein that is different in fast and slow motor neurons, and so it fits very well. So this is a way, if this change in mRNA is translated into change in protein, this is an explanation for some of the physiological changes that occur with endurance training. We also found decreases in some of these other proteins. This SK2 channel is I guess I should explain these bars, right? This is treadmill exercise is the gray bars, voluntary wheel is the black bars, and acute exercise, 7 days only, is the white bars. This is 100% of no exercise at all. So we did find also a downregulation of this SK2, and I'm going to explain in a minute. This is a small potassium channel which underlies the after hyperpolarization amplitude. And this could point, as I'm going to show you later, this could point to a potential change in the expression of the potassium channel towards a more slow-like motor neuron. So the changes in these two proteins sort of fit with the biophysical changes that we saw. We're not sure about some of these changes. They're not very big anyway. I think physiologically, to talk about anything less than a twofold change is not probably very meaningful. But we did report in that paper that there was a decrease, and some of them pointed in the direction of some of the biophysical changes. Generally speaking, we were very disappointed with the lack of change. We were very surprised. And one of the problems, I'll show you here what the 5-HT1A that was downregulated with endurance training, what it normally does. These are the receptors on the initial segment, and this is where, of course, the action potentials are generated during movement. When serotonin is released, it excites the motor neuron by binding with these excitatory receptors. These are inhibitory receptors, and they don't respond very much unless you have a buildup of serotonin. And with a buildup of serotonin, you have these receptors excited, but these ones start to be inhibitory now, and it's called the spillover hypothesis. Perrier has described this as a spillover phenomenon and says that this is part of the explanation for central fatigue, which is very interesting. And this is just the after hyperpolarization. We know that in fast motor neurons, which have a fast AHP, we have, this is governed by the slow potassium channel, SK2. And when you compare that with slow motor neurons have a much slower after hyperpolarization and larger, and this limits their firing frequencies to some extent. So what we think is happening with endurance training is the SK2 is downregulated because it's being replaced by the SK3. And we didn't at the time have SK3. This wasn't popularly known as a governor of AHP. When we did this paper, it's only come out recently that there's a significant difference between muscle types in SK2 and SK3 and the motor neurons that innervate them. So, we haven't continued much with the endurance training. A little bit later, we decided to look at gene expression in another model. This is spinal cord transection. And in spinal cord transection, there's a problem with spasticity. And part of the explanation for spasticity is an upregulation of serotonin receptors, increased excitability of motor neurons, and they fire when they shouldn't fire. So a student of mine, Jeremy Chopik, who's now at DAL and University of London, decided to look at gene expression in extensor and flexor motor neurons after spinal cord injury. Now, the thing that we smartened up with on this particular study is we decided to try to identify different types of motor neurons, different pools of motor neurons. In the first study, I think a lot of the changes with endurance training were hidden by the fact that we were taking everything: flexors, extensors, fast muscle neurons, slow muscle neurons. In this particular case, at least we separated flexors from extensors. And I'll just go here. This isn't flexors and extensors. This is another study we did. But we did the injection of different fluorescent dyes into flexor and extensor muscles in the hind limb, and so that we could label them, and then on laser capture we could pick out the ones that we wanted. So here we have flexors in the green, extensors are in the purple, and on the left you can see this is a longitudinal section of the spinal cord before cutting out the motor neurons with laser capture. The right side shows after. And I'm going to go back because I wanted to show you a result. When we did this, and we did laser, and we're interested specifically in serotonin receptors and another receptor called a metabotropic glutamate receptor, which is this one. and also this potassium transporter, because these have all been implicated in spasticity after spinal cord injury. So these were rats that were spinal cord injured. 3 months later, we looked at gene expression in the motor neurons. And this is control, and shows that in fact flexors and extensors aren't really that much different in their gene expression, which was a new finding. Here, the only difference we could find between, whoops, I'm going the wrong way here. Between flexors and extensors was in this particular protein, which is the metabotropic glutamate receptor. It's higher in the extensors than it is in the flexors, and that's because of, we know now from work I'll present in a second, it's a difference between fast and slow motor neurons. But generally speaking, flexors and extensors showed the same expression of serotonin receptors, and these are all subclassifications of serotonin receptors. There's the 1A I talked about before, the inhibitory one. There's the 2A, which is the most abundant mRNA in motor neurons, the 2C, the 7, and so on. So in this particular study, we were continuing some work where we were trying to see if we could change motor neuron properties and if we could change spasticity, in fact, by passively exercising the limbs of paralyzed rats. So this is a study that Jeremy did in my lab. I had started this 15 or 20 years ago with a colleague at Drexel University in Philadelphia who was very interested in this, and we continued some of this at University of Manitoba. These rats were exercised passively for 1 hour a day after spinal cord injury, and since they're a little bit spastic, they actually, if you look at the EMG of the muscles, it actually looks like the rats are exercising because their EMG bursts occur during the cycle. It doesn't happen with control rats, but with spinal transected rats, you can actually see EMG. So it's similar to what you would see with exercise. And when we looked at these results, and this was just published a year or two ago in Journal of Neurophysiology, we found an upregulation of the serotonin 2A. This is the most, as I said, the most highly expressed serotonin receptor, the one that is really the most responsible for excitability, highly expressed after 3 months of having a spinal cord injury. Very similar to probably what happens in humans as well. Some of them are downregulated, the 2C are downregulated, not much happened to the 7. These aren't really expressed in very much abundance in motor neurons. The chloride transporter, not changed very much. The 5-HT1A and the glutamate receptor were downregulated. Now, the interest in this particular study was that we had shown before that when you passively exercise rats after spinal cord injury, you can prevent some of the biophysical changes that occur in motor neurons. So there was a sparing effect of the exercise on the motor neurons. So we wanted to see if there was that effect in gene expression. And we found, for example, interestingly enough, that the 2A is more highly expressed after spinal cord injury. That's in the extensors. And with cycling, also, you had a further increase. So as opposed to our thoughts that you might, diminish the effect of spinal cord injury, you do see an effect that goes in the opposite direction of what we thought, an increased expression of 2A. And there were some other proteins that when you add leg exercise, for example, the 5-HT7 is more highly expressed as well. And we don't know what that means. But look at the paper in Journal of Neurophysiology and see our explanation. So, excuse me, two sort of unrelated studies. Okay, so this is the last study. It's the most recent study. We've decided, we refined the technique of laser capture and gene expression to the point now where we can do it on 100 motor neurons. The first study, if you remember, was done on like 600 or 700 motor neurons. The second one was done on several hundred for the flexors and extensors. This particular study was done recently, and the soleus and the lateral gastrocnemius in the rat are different. Soleus is mostly slow muscle fibers. Lateral gastrocnemius is mostly fast muscle fibers with some FR and a few S fibers. So we decided, now that we've got the sample where we can record, or we can capture 100 motor neurons. That's enough tissue for us. So let's try, because the soleus of the rat has about 60 to 100 motor neurons. So we injected, soleus with one color, the same procedure that I just mentioned, the lateral gastroc with another color, so we could see them in the spinal cord. We use laser capture to separate those out. And I'm not expecting you to read what's on the down here. I'm going to show you some results. So here are some differences, and you can see some of them are regulated. This is normal relative gene expression percent gastric. So this is gastrocnemius, and any difference is because of differences of soleus versus gastrocnemius. So soleus is higher here, higher here, higher here, lower down here, and so on. I'm going to show you on the next slide. A lot of differences between slow and fast motor neurons. And if we pick out the ones that are highly expressed in slow motor neurons from that graph, we find that the glutamate receptor, the metabotropic glutamate receptor, it's not ionotropic, it's metabotropic. So that means that it's sensitive to ambient levels of glutamate, and it will change via G protein. It'll change something inside the cell, maybe the excitability of a particular ion channel. We found that in slow, we have a higher expression of this metabotropic glutamate receptor. This is one of the ones we found that weren't changed with endurance training. So in this particular case, hard endurance training is not changing a fast motor neuron to a slow motor neuron. The SK3, there was a recent paper that showed us, and this is why we measured this, a recent paper that showed that SK3, which is the potassium channel responsible for the after hyperpolarization and the firing frequency range of motor neurons, is isolated, is more pronounced in slow motor neurons. So we measured this in slow and fast, and sure enough, it's much higher in slow motor neurons, and it's probably responsible for the longer after hyperpolarization. And again, we didn't measure this in the endurance training study, but we think that the decrease in SK2 probably means that the SK3 is being more highly expressed. The synaptic vesicle protein, SV2A, this is a protein that's in the synaptic vesicle down at the muscle. It's a good example of, another example of why it's easier to measure mRNA than it is protein. This synaptic vesicle protein is localized to the nerve terminals of type 1 and small type 2A, and is much higher in slow motor neurons than fast. And a recent paper by Shakalakal showed that, it wasn't a very recent paper, but 2010, but showed that it may be under retrograde control from the muscle. And we didn't measure that with endurance training previously. Okay, some of the things that are higher expressed in fast motor neurons. This is a cholinergic muscarinic receptor, G-coupled, that modulates the motor neuron in a locomotor cycle. So this is a first time this has been demonstrated. This is we're going to follow up on this particular protein. The CAV1.3 is a calcium channel, voltage-regulated calcium channel. It's responsible, it's thought, for the plateau potentials, which are very important in movement. And we didn't find any change in this with endurance training. So this is a property that is a very nice property for increasing excitability of motor neurons, but it doesn't look like, again, that endurance training is changing fast to slow. And of course, as I mentioned before, the 5-HT1A receptor, it's higher in fast motor neurons, and as you remember, we found that it was downregulated with endurance training. So this is one of the proteins that is consistent with a change in some of the properties of fast motor neurons towards slow motor neurons as you endurance train. Okay, so a couple of take-home messages here. We found that laser capture microdissection combined with PCR is very useful in giving us some ideas of which genes are stimulated by interventions like exercise training and spinal cord injury, and looking at different muscle types. Motor neurons seem to respond to spinal cord injury, exercise training, and innervated muscle type by changes or differences in gene expression. And there's no clear evidence that exercise training promotes a transition of motor neurons towards a more slow phenotype. And that's everybody that's my colleagues at the Spinal Cord Research Center. And on the bottom are the people that do all the work and coach me on how to give a talk like this. And our motto is you can't fix something that's broken, like a spinal cord injury, if you don't know how it works. Thank you.